So today we're going to get into a little bit on metabolism and growth requirements. This will be a 2-day lecture. There's a lot of material in here. So over the next couple lectures, we'll cover this. But again, it's kind of building on what I tried to convince you last time, that pathways were important. And then knowing the enzymes that the bacteria have in those pathways. So what they can produce, then that kind of informs us on how we need to grow them. Some species are a little more fussy than others. And that's the example we have in this scenario, this case study, where they had a sick infant with some particular symptoms that looked like meningitis. And so what they did is they actually did a spinal tap. So they took out fluid from the individual, put it under a microscope, looked at it. They saw there's a particular type of bacteria. But then what they did is they tried to grow it on a couple of different plates. And we're going to talk about different types of plates that you can use. But the ones that they used were mainly for what are called fastidious. I always think of kind of fussy. They need specific ingredients to kind of help them grow, like growth. They have growth factors in some of these plates, which help some of these bacteria grow. So by starting with plates where you're trying to grow just anything, but then you can also use specialized plates where you're trying to narrow down what kind of organism you're trying to grow, then that's going to tell us about what type of requirements these microbes need in order to actually grow them. And then kind of the other point is right here. Why? Why do we even do this? Maybe I'll ask you toward the end of class. I'll ask you this question again. But part of this idea is to be able to grow bacteria and also get what are called pure cultures. So I want you to be thinking about why we're doing this, what's the point, and how does it help us? And even it goes back to the germ theory. What's the point of being able to grow bacteria properly in the lab? Okay. So this particular individual, they grew it on a couple of different plates. One of them actually had red blood cells in it. So you can buy plates like this, and this will tell you if bacteria have what are called hemolysins, which will break up red blood cells. Based on how they grew and what you needed to actually give the bacterium in the plates, then they were able to narrow it down to a particular bacterium, this Haemophilus, which causes different types of meningitis. So treated him with antibiotics. Thankfully, was able to get rid of them. So that's just a nice thing. So meningitis strains can be pretty dangerous because they get into your blood, your spinal fluid, and it affects your brain. So it's pretty crazy. Okay, so we're going to talk about pure culture. We're going to talk about the different types of plates and some of the things you can use to use this for. So first question is, why pure cultures? Why is it useful to be able to grow something as a pure culture in the lab? Yes, ma'am. Right, very good. So you can only study that one organism and not other factors. So sometimes you can have natural bacteria, right, as part of your body, just say your body. And so if you have mixtures, how does that kind of affect your experiments? If you have a mixture of bacteria growing in plate or your tube, whatever. Yeah, treat the wrong. Just think of any experiment, right? Usually like to have one, what's called variable. That's the word. One variable you're working with, not multiple, and then you may end up treating something incorrectly. So peer cultures, these are important. I'll just say they're important for your experiments, whatever you're doing in the lab. You want to try to make sure of what you're growing. You don't have a mixture. And then, of course, whatever conclusions you come to, that is due to that particular organism. Okay. So let's talk about media. It's the common term that's plural, right? So medium, this is typically how you grow things. You can grow things in a broth or an agar. So broth, just want to remember, is kind of like the liquid form. So you could grow that in a tube. This shows an auger, but you can imagine a tube with liquid. Anybody ever do this? Inoculate something in the lab and grow it? So what's cool is it's mostly water, but then you can change whatever ingredients you want in that water, as long as it dissolves, I guess. So you can put pretty much anything you want in that water. The difference between broth and agar, this one has, it's like a solid media, so not a liquid. You add agarose or agar to the plate, and then it forms something like a kind of a jello type mold. So it's still mostly water, because you still need water in there, but it creates this solid base for which then you can streak different microbes on there. And then the microbes have access to the liquid and the ingredients inside that liquid. And again, you can change those ingredients for whatever you're trying to do. And then there's different things you can do. So I think in the lab, we're mainly going to be dealing with petri dishes, maybe sometimes tubes or slants like this. So this is still an agar. I don't know if we do any stabs. Later on, we'll talk about stabs are useful for growing anaerobes, so bacteria that don't like air very much. But it doesn't really matter. All of these kind of provide the same method to be able to grow things. You guys will learn how to streak microbes out. This is not that kind of streaking like you're running across a football field, okay? Don't get me wrong. This is, again, where we're trying to get a pure culture. When you can pick up bacteria or whatever. Again, it's not always just bacteria. Whatever microbes you're working with, you can pick them up with a loop like this, and then you lightly drag them along the surface of this media, and I mean lightly. It's very easy to dig down into it because it's kind of jello-like, right? Okay. But as you're doing this, and as you carry them across, you're actually making a dilution. You're spreading them out on the plate, so you're diluting the colonies, sorry, the cells, and then the cells will grow what's called a colony. So if you're talking about a plate, something like that, you streak them out. You let it grow for a day or two, depending on the bacteria. And then the cells, I guess it's kind of an assumption. We assume that individual cells will grow into what's called a colony forming unit. Colony forming unit, or a CFU. I think I have another plate. Oh yeah, this is kind of what colonies could look like on a plate. So you can, you'll see this mass of cells, which we assume comes from one cell. And as they divide, they grow into millions and millions of cells in this little mass, and that's a colony. And then depending on the microbe, you can get different shapes. So yeah, you don't need to memorize this. Okay, so just look at it. So there's different shapes you can get, but again, here you're getting information on the type of colony that it looks like. And this can vary. Or the color can vary as well. So you can get different information about whether your culture is pure. If you think about a culture that may be impure, you might see colonies that look different shapes or sizes or colors. And then that can inform you whether it's a pure culture. Isn't that kind of cool? So you guys, I'm sure you're going to do some streaking in the lab, okay, with bacteria first. Sorry, that's a dumb joke, but you know, it's kind of fun. You just don't call your parents, right? Okay. So you'll streak them out, and notice part of this plate is really thick, right? Can't really tell what's going on there. So on your side, it's way over on my left over there, okay? But as you continue to streak, and this is called a quadrant streak, so they streak them out, they get progressively kind of more dilute. And so over here, you could see the individual colonies. And those individual colonies then help you identify or give you information about the culture. Okay. So far, so good. So we're growing bacteria in lab. We're able to get pure cultures and try to isolate that to get information. Another way you can get pure cultures is to do a spread plate technique. This is also called a serial dilution. So you can do a plate like that, or you can do a serial dilution. is another way to isolate the colonies, kind of see them. This one, what you do is you take an unknown bacterial solution and you take it progressively through a series of serial dilutions. So each of these is a known volume, and you transfer the same volume from tube to tube. And if you can imagine, if that is sterile broth, then we're diluting out the number of bacteria, and then we grow those on plates, and you can actually count them. This is useful if you're trying to get a rough estimate of how many bacteria you have in here per like milliliter, then that can inform you on how much antibiotic or something like that you need to have a dose for to treat that. Okay, that's called a serial dilution. Okay, what kind of media can we have? So we can have broth or agar. Okay, so you can use both or either. You can have a minimal media, so it can be minimal, or another term for this is synthetic or defined. These are all kind of used in the same manner. So a minimal media is kind of what it sounds like. You have a minimum number of ingredients, but it's also defined. Typically, you know the exact ingredients that you want to use, and you can limit it. So then you can figure out if that bacteria can grow or not. Okay, so here's one of those things where I'm trying to figure out the growth requirements of my microbe, whatever it is. Okay. Those you typically weigh out particular salts or sugars and things, and you know exactly what it is into the liquid. Or you can have there's a couple of terms again here. You can have rich media or complex or enriched. So I'll just say a rich media or complex. Those are the two that I typically use. So here, you add ingredients to the media, but you don't really know the exact amounts of every single ingredient. So here, I'll give you an example in this one. It could be something like yeast extract. Have you ever seen this on an ingredient list? Or what do they call it? Like a beef, peptone, like a peptone digest. Sometimes you eat food, you can see yeast extract out. Yeast is a rich source of ingredients. They take yeast. You know what an extractive is, don't you? Like an extract, like peppermint oil, right? They extract something of those ingredients. Well, here, what these things are is they extract all the goodies from these organisms. So like yeast, they're full of sugars and amino acids and proteins and all sorts of stuff. They extract that, and then when you add that to food, then that's basically a source of nutrition, a wide variety of a source of things. Okay, same thing with like a peptone, or you find, I can't think of the word, but it's like beef extract or something like that. Again, here what we're doing is you're getting a complex mixture of stuff, but it's really rich. You have a lot of goodies in there, which will then help those organisms grow. And so here's where you're trying to grow. Maybe you don't know exactly what you're trying to isolate, but you're using a media which will be able to support lots of different bacteria. Does that make sense? But you don't really know the exact ingredients. You just know there's lots of goodies in there. So you're trying to grow these fussy type bacteria. So I asked that class, that son. Does that make any sense here? So with something that's defined, I know it's 1 gram of this, 1 gram of that, 1 gram of this. With complex, I just add 1 gram of like beef extract. And I know there's lots of good stuff in there. Yeah. Okay. So it depends on what you're trying to do. And I think we'll do both. I think we'll have examples of both in lab. Okay. I do want you to know these terms. So if you see these terms, then you want to kind of know what those plates are examples of. Okay. Sorry. Okay. It gets a little more complicated because you can also have selective media and differential media. I'm going to leave that up there. So in addition to these, you can have selective versus differential. Might be straightforward. I don't know. Selective media selects for a particular type of organism. And usually what it is you're adding ingredients. So this is typically a defined media. But you're adding ingredients that will favor the growth of one particular kind. So you can have media. That's one is banging on. You can have media that you can select for, say, gram positive or gram negative simply by adding a particular compound. Okay. Differential, this one is where you're really getting down to the nitty-gritty and you want to differentiate between, say, two strains of the same species. So like 2 E. coli or something like that. Okay. So selective, you select for one, I'll just say particular kind of microbe. And here, let's say you want to differentiate between 2 types. Let's say differentiate. Sorry if I'm being a little messy on the board today. Differentiate between 2 E. coli, for example. So it's the same species of bacteria, but you want to see if they have any other differences, say, in their metabolism. Or with E. coli, a lot of the time we're differentiating between a virulent and a non-virulent strain of E. coli. Because some E. coli are just fine, they don't cause us any problems. Some E. coli can kill you, right? Seen it in the news probably in your lifetime. Something's been contaminated with an E. coli, and it turns out to be a hemolytic E. coli, which can kill your red blood cells, basically, right, with hemolysis. Just trying to give you examples. So here, I do want you to know these terms. So again, we're different types of media, but now we're selecting for a particular microbe. And also, sometimes you can differentiate between. So here's an example, McConkey medium. So what does it say? It says McConkey's has salt in there, which prevent the growth of things other than gram negative. So you're selecting for gram negative bacteria. Yeah? And how do they figure this out? Probably through trial and error. Adding different things to the media and then seeing what grows, right? So that's what you get. You do that and you get something named after you, right? I'm pretty sure McConkey was the one who did it, okay? So then what is the other one? Now you can do a differential and you can do it in the same plate, right? Realize you can have this in the same plates. But this one is looking to see if they can ferment lactose or not. Okay. What does that have to do with? Well, what we talked about last time, right? Does it have the enzymes necessary to do the fermentation or not? So there you can differentiate between 2 strains of the same bacteria. Okay, does that make sense? Okay, so you can do broth or agar. You can have minimal or rich media. And then your media can also be selective and or differential, depending on.
So, selective or differential both define?
Yes, typically because you're adding specific ingredients to test their ability to do something. Yeah. So I would argue, yes, these are probably defined because you're adding a particular salt or you're adding a particular sugar, for example, to do that. Good question. Does that make sense? So yeah, they would probably fall under the defined. And yeah, if we look up, and I'm actually on Wednesday, I'm gonna hand out a little handout where you guys can look up a couple of different plates and write down information about what they do. So you can kind of go see, and you can probably look up the ingredients that you put into the plates as well. Not that you'll have to make your own plates or anything. So darn, you guys don't get to make your own plates. Kind of fun, actually. using an autoclave. You guys know how do we use them? How do we do it? I guess I should have put in a slide on this. So you mix the water and your ingredients, and then you typically put it on to, not onto the plates, but in a jar, glass jar. And you put it in something called an autoclave. You guys ever used an autoclave? At work or something? So an autoclave is a pressurized chamber. Sorry, I should have a picture of it. Where it uses steam and pressure, so heat and pressure to sterilize a liquid. And the why it's so nice is because of the pressure, it can bring up a fluid up to something like 120 degrees Celsius. So above its boiling point. So it doesn't boil over in the chamber, but using that pressure and then using such a high heat that's going to really sterilize the liquid, isn't it? 100 like above boiling. That's going to be pretty good. So then what do? You cool it down. And depending on whether it's a broth or an auger, usually the auger, then you pour it into the plates or the tubes or whatever you have. Of course, I added auger to it as well. And so then it'll solidify as it cools down. The liquid obviously just stays liquid. So you guys really we should make you make stuff in lab. So it'd be fun. Okay, let me ask you a question in here. Wake you up. Micro class. Your favorite media contains a defined amount of ingredients. What is it? Let's see this here. Who's awake and alert? You guys aren't awake, then we can do some jumping jacks. Did I get everybody? Ooh, you have an electronic thing? I didn't even think of that. Huh? Oh, that's right. You did the first day. Okay, what do you guys think? So I put a key word in there, didn't I? Defined. So if it's defined, then it's going to be synthetic. So synthetic tells you, you know exactly what you're adding to the list. Okay. Cool. So yes, I want you to know the terms, right? What's a broth? What's an auger? What is this versus that? And then I'll give you some more examples in selective and differential as we go through the class. So you'll see it over and over. Questions on that? Nothing. Okay, kids are quiet today. Okay, here's an example of where they used a urine culture. So we basically take liquid from a person. So I guess it's kind of like a spinal tap, right? You're going to use that liquid. Then usually what they do is they can plate it directly, or you can do a serial dilution or something like that to then grow whatever bacteria are from there. And I guess urine typically has a really low number of bacteria per mil, so fewer than 100,000 or 10,000. So if you get something larger than that number, then that tells you that you might have some sort of an issue if you have a lot of cells, more than you expect, right? You might have an infection like a UTI. Okay. What's the nice thing about doing this on plates versus just using a microscope? Because you can count stuff under a microscope as well, can't you? What do you think the difference is by growing it on a plate? You don't need a microscope, yeah. Oh yeah, that's a good point. So if you stain them, you're killing them, and you're on the right track. When we do things on a plate, they have to be alive, don't they, to grow? Yeah, that was a dumb question, right? They do. They have to be alive. Under a microscope, with or without staining under a microscope, you're not sure whether they're dead or alive, right? So that's actually a good point of growing things on a plate, is that you know that they're living and you don't have to overestimate how much you have there. Okay. So there is a point that you can use things under the plate. Sometimes it is nice to look under a microscope. I think last time I showed you the malarial parasite grows inside cells. So sometimes it's nice to use that under a microscope, of course. Okay, any other questions on plating, plates, plate types? I'll give you a handout next time. You guys can go do some research on that. Nothing? Okay. Is it okay then if I erase some of this stuff? Look, I'm gonna leave our objectives up there, because again, I want you to be thinking about why we're doing this. Why does it matter? Why is it cool to be able to differentiate between things? And what does it have to do with the enzymes that are in those cells as well? Okay. Let's talk about the growth cycle. There's different stages of growth that cells go through. And this is true whether it's in a liquid media or a plate, an agar media. Of course, cells they divide through binary fission. So literally that means to divide in half. Most of them go through this. We'll talk about this later on as well when we talk about genetics and how cells divide. Under perfect conditions, cells will undergo what's called exponential or logarithmic growth. So for most bacteria that's in a range of temperatures, and this goes back to the old adage, right? Don't eat stuff at a picnic that's been sitting out for hours at room temperature, right? What's the problem there? Temperature? Yeah, most of them like kind of warmish or neutral temperatures, right? So you're supposed to keep cold foods cold or hot foods hot for a reason. And I have to tell you, after teaching this class for so long, I always do a double take when eating things at a picnic and try to figure out how long things have been sitting out. What's the chances that they're contaminated? Because in the lab, at least, bacteria, they divide every 20 minutes under perfect nutrition, perfect temperature. So something like E. coli can divide really, really fast, really quickly. Okay. What are the phases of growth? So you have the lag phase, the log phase, and then you have stationary, and then the death phase. Okay. And this, again, this applies to any type of bacteria growing, whether it's liquid or solid. So the lag phase, see at the beginning, there's really not that much of a difference in the growth number. And really what they're doing here is they're kind of adapting to their new situation. So you'll see this anytime a microbe is introduced into a new environment. They always have kind of a lag phase where they're actually changing their gene expression typically to then utilize whatever's in the new environment. And then at some point, they will start to grow very rapidly. So the log phase, think about it. What's going on in the log phase? You think this is like preparation to grow? What's going on in the log phase? Okay, let's do parent share. Turn to your partners, slap each other a few times across the sheets. Okay. Reintroduce yourself if you need to. Don't make you get up and do jumping jack. What's going on in log phase? Think about these cells. Come on. Thank you for letting me pick.
Oh, of course.
Okay. Wants to report, so it comes to the over here. Wants to report. Pair share reports, right? Supposed to talk about what.
It's like has to be between road.
Yeah, yeah, but what's going on? What's going on?
So the double ways.
Sorry, Dave.
It's in like it started to eat media.
Yeah, yeah,
Fine.
yeah.
It's lots of.
What does that take? You guys already kind of know about dividing a little bit? What does that take? What did page?
It takes some.
Right, right. So they're actively eating and growing, right? So they're part of that is making cell walls, membranes, proteins, organ. Well, no, they don't have organelles. Well, unless they're a eukaryote, I guess. Depends on if they're a prokaryote or eukaryote, right? But yeah, they're actively making cells. They have to grow first in order to divide. You can't be too small of a cell to divide. So you want to think they're using our ingredients that we've provided them, right? Which means then what happens kind of in the stationary phase? So they're growing. Think about growing, dividing, using nutrients, dividing, yeah. Using nutrients in the media that we provided. What happens then in the stationary? They certainly have stopped growing. So the scale is number of cells, right? Number of microbes. So at some point, what's going to happen to the amount of nutrients in the media? Yeah, so they either start to drop, or sometimes we see bacteria that will affect each other, kind of like an overcrowding effect called quorum sensing. That's what it's called. Yeah. So usually though, nutrients start to run out. And the amount of division starts to drop off, and then cells, they start to actually die. They start to die off in this phase, okay? So you want to think about things like nutrient limitations, or I'll just put quorum sensing. You can look this up later on, where bacteria can actually affect the growth of other cells, so they can slow them down, okay? But then things start to actually start dying, right? So sometimes what they say is you have an equilibrium between division and death. So the amount of dividing kind of equals the amount of cells that start to die off. Okay. And then what happens in the death phase? Right, the amount of death has taken over, right? So more cells are dying after this, okay? So in order to keep them growing, typically what people do is they have what's called a continuous culture, where you're constantly adding fresh media to your culture this class. And then that's kind of keeping them either at log phase or stationary phase. So you're constantly providing new nutrients, and you're also getting rid of waste or other things which could negatively affect the growth of those cells. And here, this slide is actually alluding it to our bodies. Our bodies are kind of like a continuous culture because we're continuously eating, aren't we? Athletes eat a lot more than the rest of us, right? But you're feeding your culture, your microbiome, and then you're eliminating waste out the other side, and then you eat some more. So it's kind of the same thing that we see in our bodies, okay? So, yes, I want you to know the steps, but I also want you to think about what's going on during these steps, okay? So here's a typical question I could ask you guys. What stage of growth do you think antibiotics would work the best? Now, there's a better answer of these answers, right? So you want to try to think of what's the best answer, even if it's not a perfect answer. Think about it for a second. So if you're trying to affect the growth of these things, I don't give you too many hints. I want to see if you guys can get this. What do you think? What do you think? This is a good thought question. good thought questions. That's cool. I gotta remember next semester to help you guys stay in it. It's a good idea. I didn't even think of using an iPad or something like that. It's a great idea. Did I get everybody? What's the problem with lag phase? Think of it now, apply this to sickness. How about that? Where do symptoms start to show up? Is it lag phase? Well, or not, there's not a lot of bacteria around, right? Or let's just say it's a bacteria. So where do you think symptoms start when they start affecting the body more? Log phase, probably. That's where you're getting a lot more, and here it can be bacteria or virus, right? More virus infects more cells like epithelial cells. Then you start getting the sniffles when you got influenza or something, right? Okay. But then it's stationary. I mean, death, it's not death, right? They're already dying because you gave them antibiotics, for example. So where do you think the best stage for this kind of answer? What's the split, huh? We got a lot of people say lag or log. Why is it log phase? Think about this for a second. Why is this the best thing? And you may or may not know this, so it's okay. They're active, and then what do most antibiotics do? I can't talk today. Sorry.
Competitive inhibition.
Right, yeah. So they inhibit what? Growth. And there's lots of different examples. So I think what you're talking about is something like penicillin, which interferes with cell wall synthesis. And we'll learn about this. So most antibiotics, she said, competitive inhibition, talking about inhibitors. If you remember what they do, they interfere with processes in the cell. And that's why I dwelled on what's going on in log phase. In order to divide, they have to be able to synthesize all these things. So what do a lot of antibiotics do? They interfere, or antivirals. Antivirals interfere with the synthesis of new baby viruses called virions. Does that make sense? So I could see where you say lag phase would be the best, right? Before they start growing, but how often do we know they're growing before we know they're growing? That's the problem with lag phase. So log phase, you start to see symptoms, and that's where you got to start taking those drugs to interfere with that. Yes, ma'am.
So is there a stationary phase or does it restrict to death?
Yeah, probably just, I mean, death phase just means the death rate is more than the division rate. So probably with antibiotics, it just forces them out of stationary. Even if maybe they don't even get to stationary, right? They could be in log and then you just start killing them off, right? So they could skip. So it's not like they have to go. It's not like they take a vote in the culture media and say, we're going to stationary now, right? They're just doing their thing, right? So that's a great question. So with antibiotics, well, at least you hope so. If they're effective, you hope they start killing them off. And then as your doctor says, you need to finish your course of antibiotics, even if you start feeling better, right? Because you need to make sure you kill them off. Yeah. Isn't that cool? So a nice thought question. I like those kind of questions. What do you guys think? Interesting? No? Okay. Any questions on that? I think that's the last question for today. What do we got? We got a few more minutes. I can start the next one. So what do you need to know? You should know the stages. We will apply these stages to illness as well, although we just did it, right? When you first get infected, they go into lag and they start, preparing in your body to divide. And then they go into log phase. That's typically where you start seeing, why do we start seeing symptoms in log phase? We can go deeper into this. What's reacting? Way over there. Immune system, right? So in lag phase, you might not have enough foreign particles to start telling your body what's going on. But as they start dividing, now your immune system becomes more aware. Okay, we have a problem. So that's when you start you start presenting an immune response. And those are symptoms, right? Runny nose, fever, or inflammation if it's a cut, something like that. Right? Those are all immune responses, part of the innate immunity. And then hope later on you might get adaptive. Yes, sir.
So in terms of an illness, the lag phase would be the incubation phase.
Yeah. Yeah. Very good. That's another way to say it. So then you start getting symptoms there, and then either your immune system fights it off, which typically that's usually with viruses, that's the only thing that fights them off, isn't it? We don't have antibiotics toward viruses. So either your immune system does it or you take drugs to kill them off. Yeah. I mean,
So I had strep throat and I took antibiotics, and then the next day at work, I got exposed to strep again.
oh,
And
okay.
I got strep again. The
Oh.
PA told me that if I had been on antibiotics for one more day, I would have been okay. So does that mean that it can like work in the lag phase?
Well, yeah. I mean, they can work in the leg phase because the leg is they're growing, they're adjusting their gene expression. But if you take antibiotics in the leg, then it should affect their ability to go into lock, right?
Okay.
Because it's going to interfere with their growth. And I'm not a real doctor, but it sounds like something was there. Maybe there wasn't enough antibiotic or something to keep them from going, and maybe that's why it got. I'm not sure, but something was going on
Kid
there.
was strep.
Oh. Isn't working with sick people just wonderful? Yeah, it's a wonderful career. Did you have a question?
Yeah, so does bacteria go through this growth cycle even without antibiotics? System will die?
Yeah. So even without antibiotics, let's say let's just say you had a vaccine or something against it, then your immune system will make antibodies. That's the word. They'll make antibodies, which then will fight them off with the help of things like macrophages. Yeah. So I was just using an example of antibiotics forcing it into death phase, but it can do it naturally as well, right? Good question. It's great. I love these questions. You guys are doing great.
How we continue to get more nutrients and not have as much waste?
Yeah, continuous culture is a way to avoid the death phase, basically. So with this, you're instead of just having a closed system where your culture is going to run out of goodies after a while because they use them all up, you're basically diluting out the waste and you're adding new nutrients to it continuously. So what's shown here, because I can make it a bigger one, sorry. What's shown here is you're introducing fresh medium all the time to this, and then you're letting out waste at the same time. So the cells in here, you're going to try to keep them like in a stationary phase, really. Maybe log or stationary. Does that help? So I don't really use it that much because you can actually take these and just throw them in the refrigerator. And then that'll keep some of the cells from dying off. So then you can continue to use that for future cultures. But if you're studying fermentation, or this is another way to make things like this is what they use in biotechnology. So if you're making things like growth hormones or insulin or things like that, this is a way to make metabolic products. And you collect them out of here, you collect them and then purify them. So that's another way to use continuous culture. It's a great question. I won't necessarily test you on this, but it might come up on something. And we won't do it in the lab. Great, I love it. You guys are digging this. Okay, so when we come back, we're going to break it down a little even more. So we'll talk about the nutrients and growth factors. And we've got more terminology. More terminology coming up, okay? So let's see, last time we were talking about the growth cycle, how we could grow organisms on plates, right? And why is that beneficial? Why is it beneficial to be able to grow something on a plate? What is that term that we use? Okay, sorry, why is it important?
Isolate it.
Right, isolate. Yeah, pure cultures, that's what I was kind of thinking about, right? So here, we want to get a pure culture, and that's going to be important because then we know what we're testing, right? We've isolated them, we know that. Then we can test, okay? Of course, depending on the organism, the nutrients that are required might differ, but for the most part, they're going to need the same kind of nutrients as any kind of organism. Some are going to have specific requirements. So we call these guys fastidious or fussy. I always just remember fussy because we all know somebody who's fussy, right? They need special treatment for whatever they are, okay? So the same thing with some of these organisms, they might have special needs or special growth factors or special vitamins or minerals that they need. And this is kind of found out through trial and error. People try to understand what these things need to be able to grow. And then again, we can change these, right? So when we do a defined media, we can change the actual ingredients to then help us grow whatever we're trying to grow. So it can differ depending on what the organism comes, what it depends on, basically. Okay. And again, this helps us. We can understand what they are, we can understand what they need, and this can actually teach us a little bit about, okay, maybe these organisms are lacking a particular pathway or the ability to use a particular sugar, whereas other organisms can use those foods. So it really helps us kind of understand what they are and help us classify them, okay? Cool, cool. So a couple of things we want to think about. So this is like if they need special ingredients, right? Special. I'll just put special ingredients. But every other organism basically needs sources of a few particular things. Water, of course, is one of them. We'll come back to that. But you want, in terms of categorizing the types of energy needs and carbon needs, we can use some terms to help us do this, basically. So for each classification, it's going to depend on their carbon source and their energy source, their ultimate energy source, depending on what they're going to use. Okay, so we'll talk a little bit about this, but in terms of carbon compounds, autotrophs, those are the ones that are going to be able to make their own carbon compounds. Okay, so we call these auto. I'm just going to put the prefix. And on the little thing that I've handed out, I whited out a few of the boxes. So you can go home tonight and start filling them in. I'm not going to collect this one, okay? So this will be kind of a study guide. There's a lot of terminology here, but in simplest terms, the auto for carbon source, autotrophs, they're going to use CO2 to build things, basically, okay? To build things. So what's the classic example of these, of autotrophs? Plants, right? So plants sit in the sun all day long and they take up CO2 and then they build things. They build things with them, okay? And then humans and other animals come along, and what are we? We're heterotrophs, so we eat the plants and steal their carbon, or eat the animals that eat the plants, right? And without plants, we're all dead, basically. Without green plants, yeah, because heterotrophs require that, okay? So for carbon, you want to think auto versus hetero. These ones are going to use organic compounds. Organic just means that carbon containing. I'm not talking about other things. Okay, so they obtain carbon compounds from other organisms. From others. Okay, why do we need carbon? Yeah, we're carbon based. The macromolecules that we reviewed, we did review them in this class, didn't we? They're all carbon bound, or they all have a carbon backbone. So you need carbon to build it. Okay. So far, so good. Yeah, okay. So this graph just kind of shows a little bit about how they obtain their carbon. You don't need to memorize this. but they can obtain their carbon in different ways and then use it to build other things, okay? So you can kind of go back and look at this a little bit if you want to. What about energy? Photo is an easy one, yeah? And chemo. What does photo refer to? Light, okay? So phototrophs use light as energy. It's pretty easy. And then what about the chemo? What does that kind of remind you of, chemo? Well, not cancer, I guess, but, well, yeah, cancer, but what's chemotherapy? Well, radiation, what's chemotherapy? It's just chemicals, right? Chemicals. So that's what I was getting at. Chemo reminds me of chemicals, okay? So here, chemotrophs use some sort of chemical reactions, and there's different types, right? So lithotrophs can use inorganic compounds. Those are those smelly things that you smell at like the hot springs. They use minerals for their energy, like sulfur or methane, things like that. So lithotrophs do inorganic, organotrophs use organic molecules. But the basic idea here is that they're going to use either inorganic or organic compounds to get their electrons, basically. Okay, so I'll just say use inorganic or organic compounds instead of light for their energy. And some can use both, right? Some bacteria will use organic compounds for both carbon and energy. And that's kind of what we do. We are chemoorganotrophs because we take in sugars. Most people like sugars. Our cells like sugars. And we can break those sugars apart and use them for both energy, electrons, and carbon. So let's put them together then, okay? So look at your piece of paper. Does everybody have one? Let's use one. What's the first one? Photolitho-auto. Ugh, that's a mouthful. So what do we do when you're presented with a word like this and I say, tell me what energy compound they use and tell me what carbon compound? What are we gonna do? What energy and what carbon? Yeah.
Probably just break it down.
Break it down. Photo, right? So start with photo. What do you say with photo? Light for energy, right? And then it says litho-auto. Well, okay. So auto tells you what? Uses CO2, right? For its building blocks, okay? And then the litho tells you that it's they like inorganic compounds. So that's what it says in the third column. You just go across, carbon source, CO2. And the auto told us that. Energy source, light. The photo tells us that, right? And then the litho and the chemo can be kind of confusing. So you just remember those are referring to the actual chemical compound that they're using, okay? So what's going to happen on the exam? I'll either give you a word, make you define it in terms of energy source and carbon source, or I'll do the other way around. Say, what do you call an organism that uses whatever uses organic compounds for their carbon source and inorganic compounds for their energy source. So then you use the prefixes to say that's a chemoautotroph. You use whatever information you have on the question. So if it doesn't tell you like inorganic or organic, then you don't have to worry about litho. Does that make sense? Come on, you can sound smart to your roommates, right? I know what a hemolithoautotroph is. Whoa, I want to change my major to biology, right? You guys love it? You love the torture? I mean, you love the material? Come on. Okay, any questions on the expectations there? There's another table you can use. It's pretty straightforward, a little bit of memorization. Okie doke. What about some of these other things? Bacteria or microbes are commonly found in, what do you call them? Not energy cycles, but like nutrient cycles. So nitrogen's a really cool one. Nitrogen gas, you guys remember the covalent bond between these from chemistry? It's a triple covalent bond, really, really hard to break. What happens when we breathe in nitrogen gas? That's not the one that we fall asleep. That's nitrous. Nitrous. In the dentist's office, right? Well, at least they used to do that. Do they still do that? I know I had a lot of problems with cavities when I was a kid. Sorry, too much information. We just breathe it in and breathe it out. Does nothing. It's what, 60 or 70% of the air or something like that. It's pretty high. We can't even use it. But what about bacteria? There are bacterial fixers. So mostly in the soil. Anybody who's ever had a garden, they tell you to plant beans or other plants like that because they have a lot of bacteria that grow. I can't remember the term for those little nodules that are on the roots. What do they call them? Anybody remember? Sorry. But anyways, that's the bacteria in soil, which can take up nitrogen gas, break it down into things like ammonia or ammonium ions. And that is useful as fertilizer. So plants can use that. So there's a relationship, symbiotic relationship between these bacteria, rhizomes, right? That's what I was trying to think of, rhizomes. But you have a relationship between bacteria and the plants, and the bacteria are able to break these things and use them, whereas we can't. So that's kind of cool. And there's also the carbon cycle, right? So animals that die, they are broken back down into carbon compounds or CO2, right? And then that can be recycled as well. So microbes are commonly found in these cycles to recycle things, okay? What other considerations we need? Let's see, I'll just come over here. What other considerations, growth considerations do I want you to think about? Well, what about temperature? Is that a concern? And pressure?
Yeah.
Sure. Do most organisms have an optimal temperature they like to grow at? Yes. Right? In fact, a lot of them are body temperature, like 37 degrees, kind of weird. But some like higher temps, some like lower temps, some like to grow in acidic pH or basic pH. So Helicobacter likes acidic pH. Some, like at the bottom of the ocean, they need to live at high pressure, a high pressure source. So they're actually really hard to grow in the lab. Okay. So there are some terms that go with this that I want you to know. Okay. They're pretty straightforward. So in terms of temperature, you would call those thermophiles. So phile is to like, therm is heat. So you have hyperthermophiles or just thermophiles. You don't need to memorize the actual differences between like a hyperthermophile and thermophile. But you should know meso to me kind of means middle. So a mesophile kind of likes the middling temperatures. Cyclophiles like low temperatures. And then you can kind of do the same thing with acid. So you have acidophiles or alkalophiles. And then you have aerobes and anaerobes. What does that refer to? Yeah, so isn't that interesting? So not all organisms like to grow in the presence of oxygen. Some don't care. Those are like the facultative ones in the middle. Some don't care, but some are actually really sensitive to it, interestingly enough. They like to be in anaerobic conditions. So you should know the difference between an aerobe and an anaerobe. Aerobe, see if I can spell versus anaerobe. Okay, so there's differences in those considerations. Pressure, I'm not gonna test you on pressure differences there, okay? But you should kind of get familiar with these terms. So here is what the temperature kind of scale looks like. And I'm not gonna make you memorize the actual specific temperatures, but you should know kind of the terms and how they vary. So thermophiles like heat, psychrophiles don't. They're on opposite sides of the scale. Does that make sense? Yeah, thermophiles, they like high heat. I'm not gonna worry about the difference between hyper versus thermophile. But psychrophiles, those are low temps, and then mesos in the middle. So most organisms we look at are mesophiles in terms of temperature. The ones that like really extreme temperatures, we call them just thought of them. Extremophiles. We call them extremophiles. Anything that lives at like the extremes of temperature or pH or pressure, they're all kind of weird, and they usually fall into the archaea. So they're a little bit different than the normal bacteria. So when we look at the taxonomic scales of these, most bacteria are in a different group than the extremophiles, kind of interesting. So what you want to think about is they're going to have different structures in the cell to help them kind of deal with those extreme conditions. So we'll see a few of those examples as we go through, okay? So you can read about thermophiles, are pretty cool. There's psychrophiles, mesophiles, okay? Pressure, I mentioned that I'm not really going to test you on this, but you need to realize some organisms live at the bottom of the ocean, so they're under a different temperature and pressure. So again, they're going to be classified as extremophiles, okay? What about salt? Are there halophiles? Well, it's on the screen. So yeah, they found bacteria like up at the Salt Lake that grow in high salt conditions, which is not typical. It's not typical. So again, you want to think that some organisms like this can deal with changes in salt, okay? So a lot of these slides, you can just kind of go back and review. What kind of pH do acidophiles like? Low or acidic pH, right? Alkalophiles like high pH or basic conditions. Where are most of the ones that we deal with? Right around neutral philes, okay? So just be familiar with these terms, okay? Is that okay? Oxygen. So I already kind of mentioned this, aerobes versus anaerobes. So again, ones that really like oxygen, if you grow them in a tube like this, you'll find the aerobes right next to the air source where they like to have access to the oxygen. Okay? The other ones though that are anaerobic, they're typically going to be growing down toward the bottom, or at least growing best. So depending on the organism, if there's any oxygen at all, then they're not gonna, if they're really sensitive to it, they're not gonna grow at all. But if it's really, they just don't like it that much, they're gonna grow as far away as they can possibly from that. So really low oxygen levels. So it's kind of interesting. In order to grow these guys, do I have a picture? Oh yeah, there is an anaerobe jar. So there's the definitions and some examples. But in order to grow anaerobes, typically what they do is they grow them in these jars where I've done it a couple of different ways. One, you put in this pack which absorbs all the oxygen. Or the other simpler way I've done it is I just burn a candle in a closed container. How does that work? Yeah, right. Fire uses all the oxygen, or at least a lot of it. So you just burn a little candle. After a while, the candle will go out because the oxygen levels will fall so low. And as long as it's not open to the atmosphere, it stays like that with kind of a higher CO2 content than oxygen. So that's kind of done it. You can do it without fancy equipment. If you notice one thing though, I don't know if this one has it on here, but a lot of your microbiome also falls in anaerobic conditions. So if you think about your gut, it's typically a low oxygen environment. There's a lot of fermentation going on in there, as well as other things. So it's interesting that a lot of those organisms are anaerobic as well. Okay. Kind of cool. So questions on that? You need to know the terms associated with temperature, pH, and oxygen to describe these different organisms. That's the expectation. Up, down, sideways, thumb.
What's the middle area for the oxygen? There's aerobe and anaerobe, but what's the?
Oh, I think they were like facultative anaerobes or something, or aerotolerant anaerobes. I'm really not going to get into the nitty gritty of all these. So just if you know aerobic versus anaerobic, that's enough for me. I'm really not going to, and we're not going to memorize any particular organism and then classify. Acidophile versus alkalophile. I mean, if you see the word neutrophile, that's pretty obvious, right? They're right around neutral. Yeah, great questions. Good. I want you guys to be familiar, comfortable. These kind of things would show up like on multiple choice or something, right? So mostly just definition. Yeah, okay, cool. Okay, I know there's a lot of slides in here, so you can always go back and kind of review them. Culturing anaerobes, we talked about that. And then now we can get into how do we use this information. So obviously you can categorize organisms according to their needs, according to how they grow. And then going back to the fermentative ability, then you can also classify them based on what kind of enzymes they have. Okay. So remember, enzymes are specific for what they catalyze. The activity can be affected by these. So that's another thing to think about. You know, if I take a neutrophile and I put it into acidic conditions, why does that cell actually die? If I take normal bacteria and I heat them up to a really high temperature, why do they normally die? Damage, right? So you want to think about if you go to extremes with a normal bacteria, if you go and they're not an extremophile, then you're going to start losing things like enzymes, membranes, DNA, right? So these things are going to be destroyed. So that's kind of the other thing about why do we even have optimal conditions for all these organisms, okay? But back to enzymes, we know that a lot of enzymes require lost the word? Coenzymes, that's the word. A lot of enzymes require coenzymes. If you go back to the list of the nutrients that are listed, a lot of those are actually needed for these chemical reactions that we see in the cells, okay? So enzymes basically do everything. Here's a list of a lot of ones that are used in classifying bacteria. So we actually can look for whether a bacterium has these particular proteins. If they do or not, then we can classify them that way. Here's a lot of the cofactors they use. A lot of these are the same cofactors we use. So if you look at the side of your cereal box this morning, a lot of those things are actually coenzymes, believe it or not. Okay. So they're there to help your cells. Okay, what I want you guys to review is the classic pathways that are used in not just fermentation, but all of aerobic respiration as well. I'm going to change color so that they can look better. So, metabolic pathways. What is the main one that cells use for sugars? Sorry. I'm old, I can't hear you. For that, even. Glycolysis. Good. I'm glad you remembered one of them. That's good. So glycolysis. So remember that for at least most sugars, they're going to go through the glycolysis pathway. So if we have sugars coming in to the cell. They're going to go through glycolysis. And then you can have different pathways from that. So depending on whether you're going to go through the aerobic respiration pathway, so if oxygen is available, or if you even use oxygen. So some of these organisms don't use oxygen in order to process their energy. So aerobic is on the left over here. I'm really not going to go back and test you on parts of the pathways. Hopefully you got that in Gen Bio, or at least should have been exposed to it. I know you do in my lecture, because I love pathways. So anyways, aerobic respiration, that's the one where you get a lot of ATP per glucose, right? It's something like, if I remember correctly, it's like 36 ATP per sugar, something like that. So most people argue that's the most efficient way to go. You get a lot of energy from the least amount of sugars. But so you have to have oxygen available. If you don't do that, then you can do a lot of anaerobic respiration. And this includes a lot, when we just talked about it, like those lithotrophs, right? Lithotrophs fall under this as well. So they can use a lot of inorganic chemicals to then go through anaerobic respiration and release more energy from these ions, or at least using these ions to then survive, of course, right? Because the whole goal here is to make a lot of energy. So this is one example, I think, of like ions that do this thing. You can also do things like fermentation. So fermenters, these ones are going to use organic compounds to go through anaerobic respiration. So this would fall under the chemotroph kind of category. And there's several different types of fermentation. We'll talk about a couple of different types. So things like alcoholic fermentation, or ethanolic is another word. I guess that's a better word. And then lactic acid fermentation is another type. Okay, I'm gonna change the alcohol. Doesn't look right. Ethanol. Sorry. Ethanolic fermentation. So again, here you want to think, in order to use these different compounds, you have to have different enzymes involved in the pathways. So anaerobic pathways would kind of be on the right-hand side of the screen. Aerobic pathways are on the left, and they're going to use different enzymes, different proteins, and we can test them. The problem with these, though, with especially things like fermentation, you're going to get less ATP per sugar. So if we just look at the example we use in Gen Bio, it's something like just 2 ATPs per sugar instead of 36. So it would be safe to say that most of these anaerobic processes are less efficient. You get less energy per molecule of whatever they're using. So for organisms that do this, they have to ferment a lot more sugar, don't they? If you need the same amount of energy and you can't use aerobic respiration, then you're going to be burning a lot more of these other sugars. So if you look at you guys ever hear of the trypanosomes and sleeping sickness? I guess it's mostly in third world countries. But these little trypanosomes, they go around in your bloodstream. They're little freaky looking worm things. And they basically eat all the sugars in your bloodstream. So what does that mean for the rest of your cells, their sugar levels? Start to drop. So you start to get lethargic, right? Fatigue. And that's why they call it sleeping sickness, because these little buggers are just fermenting all your sugars before you're able to use them. So I should put a case study in here. I guess I will. I'll go back and put it in a case study for sleeping sickness so you guys can see it. Okay, so what are we doing here? I want you to go back, review this a little bit so that you can compare the aerobic respiration and anaerobic respiration. Here's some examples. So you have things that use like nitrate ions or sulfate or sulfur or methane, still things like that. So a lot of ones I mentioned earlier, where they're really kind of stinky little organisms in those pools, okay? Still essential for things like nitrogen and sulfur cycles, right? So these organisms are still part of recycling different compounds. Fermentation, on the other hand, so we're going to release energy from sugars. So just think like yeast, we're going to take sugars and they ferment them into CO2 and ethanol. Doesn't require oxygen, right? So we're still anaerobic. And it doesn't require a lot of the other enzymes, right? So again, we can see even with, we forget like things, somebody mentioned citric acid cycle. We don't require these enzymes from citric acid cycle in fermentation. So again, there's a difference. There's a difference in the type of proteins that you can use, okay? Okay, you guys look like you're struggling here. Let's take roll, ask you a question here. Sorry, I've been talking a lot today. So if I throw this question at you on a test, for example, what would you say? Hopefully you would answer the question. All you need is like one fingertip on that thing and it just does, it's not happy. So it is definitely a little fastidious, right? Thank you, thank you. So what did I just tell you about ATP, right? It uses more sugar to make the same amount of ATP. So in general, fermentation is going to produce less ATP per sugar, right? So these things have to do a lot more. Okay, cool. I had to do one today at least. There you go. Okay, how are we doing? So questions on these things? We got a few more minutes. Thank you for the vote of confidence there. Very nice of you. Okay, so I think I'm making my point though here. So it depends on what enzymes you have available in order for these organisms to be able to produce whatever their compounds are. So here's some examples of some differences you might see. All these are fermenting the same sugar and they start with pyruvate. But depending on the enzymes that they have, then they can make different end products from that. So go back and think about these pathways that are working together to make the final compound. And this kind of shows you what some of the differences you can have. So say the Streptococcus or Lactobacillus, we're very happy if we like yogurt, that it only makes lactic acid, right? Not some other nasty compounds that won't taste as good. Or if you like to make bread, you're happy that Saccharomyces makes CO2, right? We don't care about the ethanol part when we're doing bread, but the CO2 part is what allows your bread to rise, right? As the yeast is fermenting. So for some of these things, we're happy that they're doing that, okay? And we can use that. This is just a repeat. So this is the two types of fermentation you should be familiar with, ethanolic and lactic acid. We'll talk a little bit more about them as we go through it. But the main difference I just kind of mentioned, Lactobacillus makes only lactic acid and can be used for a lot of different fermentation products. And of course, alcoholic fermentation is the one that it's done by Saccharomyces, okay? So those two are really the only two that you need to be familiar with. Okay, let me just show you some other useful parts to this. This is a review. You can go back and review it. What can we use this for? So here was our question, right? Well, a lot of this has to do with classifying them by energy, nutrient requirements, and then fermentation tests. So another big one we use in the lab is we can do tests that will test the cells for a wide variety of enzymes, including antibiotic resistance. That's based on enzymes as well. So you could test for any of these things in the lab. Let me give you some examples. So here's some examples. You don't have to memorize any of these. Yay. Okay. But I wanted to give you a handful that I thought were kind of interesting. But think about what they're doing. So this one is based on fermentation, and it's a sugar broth that's in there. Okay. So there's two things you can look for. One is a change in color. So if you remember chemistry, color changes, a lot of those were due to pH changes. So if they're able to ferment these sugars, then you get a change in color, which could be positive. And you can also get bubbles. And if you look really closely, they actually have an inverted glass tube in there, a small one. And so you can see it actually collect bubbles of whatever the gas is, okay? So there's two things that you can look for there. This one looks for urea hydrolysis. So you put urea in there and you ask the question, does the organism break down urea? Yes or no? And a lot of these, they're a little slow. You might have to wait a day or two to get this information, but it can be helpful in identifying exactly what strain you're dealing with. This one is starch hydrolysis. So you have starch, which is colored purple in the plate. And if they break down the starch, it looks like a clearing zone in the plate right there, okay? It's kind of cool. So you can see they're actually breaking it down. And then this one is called a litmus reaction. So it actually looks for a pH change as well as this idea of proteolysis. So you actually put milk proteins in there. And if it curdles, then it tells you that the bacteria is basically fermenting or breaking down the proteins that are in the milk, okay? Come on, isn't it pretty cool colors? Colors, ooh, yeah. More colors, what do you think? Okay, I'm picking on you. Here's another one. This one's kind of cool because it looks to see whether they actually reduce iron. So really cool, it turns black. It's really easy to see the change in color there, okay? Kind of interesting. Oh, okay, so that's the payoff. That's the payoff. Is really, when you're looking at these things, you want to think about the proteins involved or the enzymes involved. Do the organisms have them? If they do, then they can do the reactions. If they don't, then they can't do those reactions. So you're classifying them based on their abilities to do different reactions, okay? That's pretty much it. Biofilms. Okay. We could talk about biofilms later, but this one was included in this chapter. Biofilms are basically just communities of cells living together. So we'll probably see more examples of those later on, but I don't think I need. Okay, questions on the expectations here? We've got some terms to learn. You need to be able to categorize organisms based on their energy and carbon needs. And then I want you to think, if I throw out a question like, why are fermentation tests so useful in the lab? You need to be able to give me a couple sentences on, well, this tells us if these organisms are capable of doing this, and then helps us classify or ID them, right? Which of course then can lead to treatment or whatever else you're trying to do, okay? Questions on that?